We're going to put all of our chemistry knowledge to a specific biological purpose in this chapter about biological macromolecules. There'll be an overview of what biological macromolecules are and why we care about them, and then how we make and degrade them. After we know the basics, we'll take a close-up look at each of the four, which are proteins, carbs, lipids, and nucleic acids. Most macromolecules are made from singular subunits or building blocks called monomers. Monomers can bind with one another using covalent bonds to form larger molecules known as polymers. In doing so, monomers release water molecules as byproducts. This type of reaction is known as a dehydration synthesis, which literally means to put together while losing water. In the dehydration synthesis reaction shown above, two molecules of glucose are linked together to form the disaccharide known as maltose. In this process, you can see the water molecule being formed. It's kind of an interesting concept because we know now that as we link monomers together in our own bodies, we actually create about 10% of the water we need in any given day just by putting together chemical reactions. Polymers can then be broken down back into those monomers in a process called hydrolysis, which means splitting water. During this process, a water molecule is actually used to break down that long chain. The polymer is broken into its two individual components when one part gains a hydrogen atom and the other part gains a hydroxide molecule. In the dehydration reaction shown here, the disaccharide maltose is broken down to form two separate glucose monomers. Note that this reaction is simply the reverse of the reaction that we saw on the last slide. It's important to remember these reactions actually don't occur on their own. They're enzymes that help this process along. Enzymes catalyze chemical reactions and cause them to happen faster. We'll talk a lot about enzymes when we get to the discussion on metabolism, and we'll learn exactly what it is that they do and how they go about doing it. So let's start our discussion of specific macromolecules with carbohydrates. All carbs have the same component, and the chemical formula for carbs is written as CH2O and then N. That N tells you just how many times that specific formula is repeated over and over. If that N was a 17, then you would have CH2O 17 times repeated. Another way of saying this is that the ratio in a carbohydrate is always 1 carbon to 2 hydrogens to 1 oxygen. The name carbohydrate actually gives you a hint as to what it contains. Carbo is from carbon, and hydrate is a reference to water, which hydrates us. All carbs fit into 3 separate groups. Monosaccharides, are singular units of carbs, also known as sugars. Disaccharides are 2 monos put together, and then polysaccharides are many units linked together because poly means many. Monosaccharides are classified based on the position of their carbonyl group, that C double bonded to an O, and then the total number of carbons they have in their backbone or down their chain. Aldoses have the carbonyl group at the end of the carbon chain. Monosaccharides known as ketoses have the carbonyl group smack dab in the middle. And then there are 3 more monosaccharide types, trioses, pentoses, and hexoses, that have 3, 5, and 6 carbons in their backbones respectively. And that little carboxyl group, as you can see, is just sticking off the end. Glucose, galactose, and fructose are all hexoses, and they're all super common monosaccharides. We have them in our bodies, they exist in plants, they're all over the place. Those 3 particular monosaccharides are actually all structural isomers. So they have the exact same chemical formula, but just a different arrangement of their atoms, and that actually excludes them from kind of trading places in a lot of biological processes. 5 and 6 carbon monosaccharides exist in an equilibrium between linear and ringed forms, so they can hop back and forth. When the ring forms, the side chain that kind of closes in and forms an extra bond to make the ring is locked into something called an alpha or beta position. The alpha or beta idea comes from a 3D understanding of structures. If you're looking at the 3D structure and the hydrogen sits behind the carbon, we call that an alpha bond. If the hydrogen is sitting in front of the carbon, then it's called a beta bond. This is an organic chemistry hallmark that allows us to predict how chemicals will bond and how they're going to react with one another It's not necessarily important for this class, but it is kind of, you'll see that a lot, the moniker alpha or beta bonds. Just know that it's a reference to organic chemistry. Fructose and ribose, those other sugars we learned about, they also formed rings, although their rings are 5-membered rather than 6-membered. It's just the number of carbons in their backbones. So they look a little bit different and perform different jobs. Disaccharides, di means two, is what you get when two monosaccharides come together after undergoing a dehydration reaction. During this process, the hydroxyl group of one monosaccharide combines with the hydrogen group of another monosaccharide, and you end up releasing water. The covalent bond that's formed is known as a glycosidic bond. Glycosidic bonds can either be the alpha or beta type, your hydrogen might move. Sucrose is an example of a disaccharide, and it is formed when a monomer of glucose and then one monomer of fructose are joined together. And of course, in this process, you lose a little bit of water. By convention, the carbon atoms of monosaccharides are numbered. And when we number the carbon atoms, it just allows us to track the way that the reactions are happening with one another. So those are the little blue numbers that you see. It allows us to remember exactly which carbons are interacting, and that gives us an idea of its overall shape after the reactions have occurred. Here's some more common disaccharides. There's maltose down there on the bottom. That's grain sugar. On the upper right is lactose. That's the sugar found in milk that some people have a hard time digesting. And sucrose is on the top left. That's your typical table sugar. There are polysaccharides, of course, really long chains of monosaccharides that have been linked by those glycosidic bonds are known as polysaccharides. Poly just means many. When you have the really long chains, they can be branched or unbranched. You see an example of both. And they can contain all sorts of different types of monosaccharides. That just doesn't have to be one long chain of glucose or one long chain of fructose. Starch, glycogen, and cellulose and chitin are really primary examples of polysaccharides that exist in great abundance in our natural world. Amylose and amylopectin, seen here, are two different forms of starch. That's what you find in a potato. Amylose is composed of unbranched chains of glucose monomers, and they're connected by alpha-1,4 glycosidic linkages. That means that carbon 1 and carbon 4 on two different glucoses are attached to one another. Amylopectin is composed of branched chains of glucose monomers, and they are actually connected by alpha-1,4 and then alpha-1,6 glycosidic linkages. You can see they're counting the carbons for you in this picture. Because of the way the subunits are joined together, the glucose chains have a helical structure. So if you don't remember, a helix is what DNA looks like. It's that curlicue. So these wind up quite tightly. There's another really common starch, and it's called glycogen. And it looks a lot like that amylopectin, but it has even more branches that are hanging off the side of it. In cellulose, common structural material in plants, the glucose monomers are linked in unbranched chains, and they have beta-1,4 glycosidic linkages. So the hydrogen is sitting in front. Because of the way that the glucose linkages are joined, every glucose molecule is actually flipped over when compared to the next one. So it results in like a really linear structure that is incredibly strong and kind of like a fiber. Insects have hard outer skeletons made of chitin. That's a type of polysaccharide. And we're going to look at all of these different polysaccharides a lot more closely as they come up in discussions over the course of the next semester. Hydrophobic lipids are in the fur of a lot of different aquatic animals, such as the little river otter here, and it helps to protect them from the elements. It keeps them warm. And lipids are going to be our second biological macromolecule up for study in this video. Lipids include a very diverse group of compounds that are largely nonpolar in nature. They're nonpolar basically because they have hydrocarbons that include nonpolar either carbon-carbon or carbon-hydrogen bonds. They do not like the charge that's on water, so they clump together and they're insoluble. We've all seen that when you try to wash olive oil out of a pan, it beads up. Lipids can perform a lot of different functions in a cell. Our bodies store energy for long-term use in the form of fats, as we all very well know. Lipids can provide insulation, keeping things like that little otter warm in the environment and do lots of other things for us. Fat molecules are types of lipids, and they are composed of two main parts. There's a glycerol seen here, and then something called fatty acids. Glycerol is just an organic compound that has 3 carbons, 5 hydrogens, and then 3 hydroxyl, the little OH groups hanging off the side. Fatty acids, however, they're a little bit different. Fatty acids have long hydrocarbon chains, and then they each have their own little carboxyl group. We know carboxyl is the C with the double bonded O, so we refer to them as fatty acids. The number of carbons in a fatty acid can range a ton. It can range from 4 to 36. The most have between like 12 and 18 carbons. In an actual molecule of fat, the fatty acids are attached to the 3 separate carbons that you see in that glycerol. We call that bond, it's a special kind of bond, and we call it an ester bond because the bond is made with the help of the oxygen atom. Triglycerol is formed by joining the 3 fatty acids to the glycerol backbone with a dehydration reaction. Hopefully, it sounds nice and familiar. We get rid of water to form the bonds in lipids, just like we get rid of water to form the bonds in carbohydrates. Everything kind of runs together. So about that ester bond formation. During the ester bond formation, 3 molecules of water are released. The 3 fatty acids in a triglycerol may be similar, or they may look quite different from one another. Fats can also be called triglycerides because of their chemical structure. That name might sound a little bit more familiar. Our doctors often talk to us about our triglyceride levels. Fatty acids may be saturated or unsaturated in nature, and in the fatty acid chain, if there were only single bonds in the neighboring carbons of the hydrocarbon chain, we say that that fatty acid is saturated because the fatty acids are saturated with hydrogen. You can't actually fit any more hydrogens onto that chain. There's nowhere for them to go. So we refer to them as saturated fatty acids. Here you see a stearic acid, and this is a really common example of a saturated fatty acid. When the hydrocarbon chain contains a double bond, we say that it is unsaturated, and that's because technically speaking it could hold more hydrogens, but it doesn't because 2 carbons are sharing their electrons, so it has fewer hydrogens than it could potentially hold, so it's unsaturated. Oleic acid is a really common example of an unsaturated fatty acid. Saturated acids have hydrocarbon chains that are connected by those single bonds. Unsaturated fatty acids have 1 or more double bonds. Each double bond may be cis or trans in configuration. You'll see this in your book. In the cis configuration, the hydrogens are on the same plane as the hydrocarbon chain. So again, we're thinking about having a 3D structure in front of us. And if everything is in one straight line, it all exists on the same plane, we could lay it out on the table and it lays flat. We call this a cis configuration. In the trans configuration, the hydrogens are actually on opposite sides. In the cis configuration, there are some double bonds that also cause a kink in the chain. So in this trans configuration, you actually have kind of a fatter, bigger, fuller shape. And that means that the cis configuration saturated fatty acids take up more space, and they're not as great at bonding, and our bodies actually aren't as good at handling them, I guess you could say. Our bodies don't know how to react to these trans configuration saturated fatty acids, and it turns out that actually means a lot of bad things for our health, but we don't quite understand the mechanism as to why that is yet. Some practical applications of lipids: the waxy covering that you feel on some leaves that's made of lipids. It allows water to run off of these leaves. Leaves have little tiny openings in them called stomata, and you don't want water pooling on leaves because it actually detracts from the leaves' ability to take in oxygen and get rid of CO2. So sometimes lipids just have a really practical purpose. We're not so much concerned with all the crazy chemistry behind them, but rather what they can do for us. A phospholipid molecule is something where we are very concerned with exactly what it can do for us. A phospholipid is a molecule with 2 fatty acids and a modified phosphate group, and it's all attached to one of those glycerol backbones. The phosphate may be modified by the addition of charged polar chemical groups, and that makes that phosphate quite differently, and the whole phospholipid acts quite differently. Two chemical groups specifically that might modify that phosphate are called choline and serine. Both choline and serine attach to the phosphate group at the position labeled R. You can see it up top, it's quite small. This allows us to identify the type of phospholipid that we might be dealing with. The phospholipid bilayer is a major component of all cellular membranes. The hydrophilic head group is the end that has the phospholipid on it, and it likes to face out into an aqueous solution. The hydrophobic tails are sequestered in that layer in the middle, and this makes for a cell that's going to look just like a ball. The outside of the ball is going to be perfectly happy dealing with water, and the inside of the ball is going to be perfectly happy dealing with water, and you're going to have a barrier around that whole cell created by those hydrophobic tails, and we're going to study the heck out of that barrier. Steroids are another kind of lipid that are important for study. Steroids such as cholesterol and cortisol are composed of fused hydrocarbon rings. We don't often think of steroids being grouped together with fats, but they do fit in the category of chemical lipids. We'll talk a lot about steroids when we learn about cellular communication and control. Proteins are our third biological macromolecule group, and they're one of the most organic molecules in living systems, and they have some really diverse ranges in function. Proteins may be structural or regulatory. They might work in transport, defense, storing things, helping us move. This big chart shows you lots of different types of proteins, examples of those proteins, and then potential functions. Basically, any science class you're ever going to take is going to harken back to proteins somehow. So let's take a look at the individual units that make them up. At the most simple level, proteins are made up of amino acids. All amino acids share this general structure. At the center of it all, there's a carbon, known as an alpha carbon, which is bonded to a single hydrogen and an R side group that varies in every amino acid. R is largely just a placeholder kind of word that tells us that that little bit can vary. Every amino acid has an amino group that hangs off the side. That's where its name comes from. And then on the opposite side is a carboxyl group. There are 20 common amino acids found in protein, each with a different R group, that's the variant group, and it's going to determine the chemical nature of that amino acid. Peptide bonds are formed between individual amino acids through a dehydration synthesis reaction. We've studied this one quite a bit. The carboxyl group of one of the amino acids is linked to the amino group on the other amino acid, and in this process, a water molecule is released. Hopefully, this looks nice and familiar. Anytime you see peptide bond, know that you're talking about a protein. There are 4 levels to protein structure, simply numbered 1 through 4, but addressed by their Latin names, and we're going to look at each of those 4 levels in turn. The primary structure is simply the chain of the amino acid, and the amino acids they can be read off one by one, just like letters in a word. The unique sequence for every protein is actually ultimately determined by the gene that codes for that protein. A change in nucleotide sequence of a gene's coding region can lead to a different amino acid being added to a growing polypeptide chain, and a single chain in amino acid can cause a complete change in protein structure and function. So the primary structure is far more important than we might be led to believe. There's some interesting examples of what we can learn from proteins when we look at the base structures very closely. Here's an example of a known and well-studied amino acid chain that has a great impact on human health. Bovine serum insulin is a protein hormone made up of 2 different peptide chains. Here you see them simply labeled A and B. In each chain, the primary structure is indicated by the little 3-letter abbreviations. It's just shortened versions of the amino acid names. And you can see that they're linked by some sulfide bonds that are interacting between some of the amino acids. 2 sulfide bonds connect these A and B chains together, and then a third actually helps the A chain fold into the correct shape. We know all these individual bonds and all these amino acids so well because we actually use bovine serum insulin to treat individuals with diabetes. If you know anyone who has diabetes, the insulin that they purchase to keep themselves healthy with a normalized blood sugar level is actually bovine serum insulin. Or another well-known amino acid chain is the one that's studied within human red blood cells. RBCs carry hemoglobin, which, among other things, allows us to stay oxygenated appropriately. The beta chain of hemoglobin is about 147 residues in length, yet a single amino acid substitution leads to a disease called sickle cell anemia. In normal hemoglobin, the amino acid at the seventh position is called glutamine, but if that gets replaced by an amino acid called valine by mistake, then that individual is said to have sickle cell hemoglobin or sickle cell disease or sickle cell anemia. In this blood smear, this is about 335 times magnification normally. You can see that the cells have a really weird crescent shape. Our red blood cells normally should kind of look like a bagel wrapped in saran wrap. There shouldn't be a hole in the middle, but you still have that depression. If you have sickle cell anemia, those individuals tend to be breathless, dizzy, they get a lot of headaches, they have a lot of stomach pain, and it's because their red cells, with these really odd shapes, not only aren't terribly capable at carrying oxygen well, but they get stuck in capillaries and joints, all sorts of places, because they just don't flow through our bodies particularly well. And this whole thing is caused by one amino acid substitution. In the 4 levels of protein structure, we're going to move now onto the secondary level, secondary structure. Those are the plates and those are the curls and the pleats within the protein itself, called alpha helices and beta pleated sheets. The local folding of a polypeptide in some regions gives rise to the secondary structure of the protein. There are 2 very common structures, again, the alpha helices as seen here and the beta pleated sheets. Both of these structures are held together by reactions between hydrogens and carbonyl groups in the given protein, and they can just either fold or curl. We'll move up now to the tertiary level of structure, and this is what happens now when you have bonds between those big alpha helice units and the beta pleated sheets, and it gives the protein its overall large structure. So that tertiary structure is actually determined by a variety of chemical reactions. These include hydrophobic regions coming together to hide from any potential water. There can even be some ionic bonding because different parts of the protein have an overall negative or overall positive charge. Hydrogen bonding will pull together a couple of things, and then there'll be some more disulfide linkages, more linkages between little sulfurs that are hanging off the sides of the protein. A lot of different kind of cards come into play when it comes to creating the tertiary structure of a protein. Here's an overall review of those 3 levels, and then we're hitting on that 4th level, which is known as the quaternary structure. So let's look at them again. The primary structure, that's just the amino acids read in their list. The secondary structure is when those amino acids start to react with one another. You have the hydrogens reacting with those C to the double bonded O. The tertiary structure is when the alpha helices and the beta pleated sheets come together and you get one big protein unit. Quaternary structure is the 4th level, and quaternary structure only exists in some proteins. This is what happens when a whole bunch of completed proteins come together to form a single functional unit. So you might have 3 or 4 proteins that are completed on their own, but they come together to form a bigger unit. So you can think about it how each of the transformers is their own transformer, but then they can come together to form the Megatron and they serve a different purpose. Proteins do that too. The final biological macromolecule that we're going to discuss are the nucleic acids. Nucleic acids carry all the information for life. They're kind of our genetic lifeblood. And there are 2 major types of nucleic acids. They're known as DNA and RNA. A nucleotide is made up of 3 different components. There's always a nitrogenous base, a pentose sugar, and then 1 or more phosphate groups depending on the nucleotide that you're looking at. The carbons, let's focus first on that pentose sugar. The carbons that reside within that pentose are numbered 1 through 5, just like we had talked about before. The base is always attached to the number 1 position of the ribose, and the phosphate is always attached to the number 5. So count out those carbons and see what you're looking at. They show you off to the right-hand side. When a polynucleotide is formed, the 5' phosphate of the incoming nucleotide is going to attach to the 3' hydroxyl group of the end of the growing chain. The two types of pentose that can be found in nucleotides do vary. We have a different sugar in DNA than we do in RNA. Deoxyribose, which is the sugar found in DNA, is really similar to the sugar found in RNA, known as ribose, but it has an H rather than an OH at the number 2 position. So about those bases I mentioned, the bases can be divided into a couple different categories. Some bases are called purines and some bases are called pyridamines. Purines are the bases that have 2 rings in their structure, and then the pyridamines have a single ring in their structure. The bases that are found in DNA are the A, T, C, and G. So adenine, thymine, cytosine, and guanine are the bases in DNA. And then RNA contains A, U, C, and G. So adenine, uracil, cytosine, and guanine. We're going to look at these bases and how they pair really closely when we look at genetics. Native DNA, normal DNA that we haven't messed with in the lab, has an anti-parallel double helix. So the phosphate backbone, when you look at the picture, that's the big blue curvy line on the outside, exists on the outside of the structure, and then all of the bases are on the inside. Each base from one strand interacts with the bases on the other strand through the hydrogens that are sticking off to the outside. So if you think of DNA as a ladder, and then you twist that ladder, the outside of your ladder are the phosphates, and then the rungs of the ladder will be made up of your bases. I want you to grab a few pieces of paper, maybe a couple like the smaller Post-its are perfect, and draw a few nucleotides. Now try and make a DNA strand with the bases pairing together and the sugars on the outside. And you're going to see where that anti-parallel structure exists rather quickly. In a double-stranded DNA molecule, the 2 strands run anti-parallel to one another because it's the only way that those bases can fit together. We label our strands with a reference to the 5' and 3' ends that are sticking out on the base. We mentioned the 5' and the 3' previously. So we say that the strands run 5' to 3' and the other strand runs 3' to 5'. All we're talking about when we say that is which carbon is sticking out. The phosphate backbone is always located on the outside, the bases are always located on the inside, and in here you can see that adenine is forming some really weak hydrogen bonds with thymine, and the guanine bases are forming weak hydrogen bonds with the cytosine. That structure, a double-ringed base and a single-ringed base coming together are really important. If you start mixing up your bases, you try to maybe bond 2 double-ringed bases together. When you get to the next pair, if they're both single-ringed, they're actually going to be too far apart. Having incorrect pairing will create bubbles in your DNA, and our bodies will notice that and actually go back and correct the issue. We'll discuss that at length when we talk about genetics. RNA structure is extremely similar to DNA structure. It's an open one-sided chain that uses uracil rather than thymine. There are 4 major types of RNA that we'll run across in the next few chapters. There's RNA that makes up ribosomes. Some RNA shares messages. There's RNA that helps to build proteins by attaching amino acids together. And then there's actually RNA that can stop proteins from being built. The RNA that composes those ribosomes is something like a conductor in a musical, and it is going to direct the DNA messages as they're being made into protein. Let's take a closer look at that rRNA, or the RNA that makes up ribosomes. So ribosomes are organelles, technically, that have 2 parts. They have a large subunit and a small subunit. Messenger RNA, RNA that's sharing messages from the nucleus of the cell, sits between those 2 subunits. tRNA molecules are actually going to fit within that big piece. They're going to recognize the codon on the mRNA. They're going to recognize the message on the mRNA, and they're going to bind to it. And it's going to cause complementary base pairing. And that tRNA is going to add amino acids to a growing peptide chain. When we discuss genetics, we're going to get to know the structure and its subunits incredibly well. So let's end our lecture with a reminder of those features of RNA and DNA. DNA is going to carry genetic information for us. It's only going to exist in the nucleus of our cell unless our cell is actively dividing. It exists as a double helix. The sugar it has is deoxyribose, so it's only got an H hanging off the end of it. Its smaller single-ringed bases are cytosine and thymine, and its bigger double-ringed bases are adenine and guanine. RNA, on the other hand, is involved in protein synthesis. It is capable of leaving the nucleus. Generally speaking, it's single-stranded. Its sugar, its pentose sugar, is called ribose. Its small pyridamine bases are cytosine and uracil, and its large double-ringed bases are adenine and guanine. To review this material, read the chapter if you haven't picked up on that already. The chapter is fantastic, and most of the information in this video can be found almost word for word, or a lot of the times word for word from the chapter. So if I go too quickly, or if you're not entirely certain as to what I'm saying, the information is in your book, so read it. Make sure you're answering the questions at the bottom of each section from in the chapter, and then check Learner for any homework assignments.